have a class, and today will be the review section of the exam 3. Okay, so first of all, lots of you are prepared the extra points, so we want to remind you again what will be the extra points for the exam 3. Okay, extra points, exam 3 will be number 1, aerobic respiration cycles. Okay, aerobic respiration cycles will be from glucose. We have this guy here, and go to pyruvate, and then go to acetyl-CoA, and then go to citric acid cycle, we call TCA cycle. Okay? So what we're going to do is the chemicals name 0.5 points each, structure 1.0 points of each. Okay, second, amino acid or side chain. And the same thing, we have the name will be 0.5 points, and the structure, because this is a little bit easy, so the structure will be 0.5 points also. Okay, number 3, what we're going to do is ATGCU, the full name, and this will be 0.5 points of each, and the structure will be 0.5 points of each also. Okay, that is the extra points for that. Besides what I said in the exam, we definitely got to test you is lac operon, the fourth scenario, which is no glucose. Excuse me, please, just don't talk there, because other people may need to listen. Okay, no glucose, no lactose. That's a scenario number 1. Then no glucose with lactose, and glucose but no lactose, and the last one section will be glucose and lactose. And we need to let you know is the RNA polymerase combined or not with promoter, and whether this transcription happens. Okay, this is the key. And whether can combine or not with activator protein, which means cap area. Okay, that will be the normal question for you. Okay, so this is extra points. Very important, please go ahead and do it. Try your best to do, even if you don't know the structure, please at least you can write some names there and some of the structures there. Now, when we do this grading, what we will do, you're missing like 1 or 2 area, it is okay. Okay, so for example, this is lots of people will make a mistake. I would have made this mistake too. So for example, fructose 1,6-biphosphate. This will be a very important. This should be hydrogen. Okay. Sometimes you may make a mistake. You write this. That's okay. That's not being we're not going to be that picky. Now these type of hydrogen and hydroxy bonds up and down. That's okay, because this is just a left, right chiral. Or sometimes those are invisible, that's fine, but you can't draw like that. That's not correct. Okay? You can draw like this. That's not correct. Which means try your best to memorization of that. If you're missing somewhere a little bit, we understand that we're still going to give you the 4 points. But if you are completely skewed, then that points will be taken off. So what we say is that I told everybody who is grading, and I'm going to grade it again, we do these things, we call it a light grading, which means the structure we're going to try your best to do. Of course, they have to be close to the correct answer, but you miss a little bit. 1 or 2 chemicals, that's okay. Okay, but the basic chemicals you have structure have to do. You can draw a triangle there or rectangle there. That's not correct. Okay, that's what we say. And 95% of them are correct. That is fine. Let's give you some of the flexible too. Made some of the mistake, but encourage you to remember it. Because some people will give up. They know the glucose is there and they're almost done. Maybe 2 or 1 or 2 structure there. Maybe forgot about hydrogen and the hydroxy bonds up and down, but then they give up. Question?
So if we're doing both the structures and the names on the exam, do they have to be like, do the names have to be right beside the structures, or can we have like a list of the names and just?
I'll give you 3 empty paper. You do whatever you like.
Okay, thank you.
Don't worry about it. You have enough space to write. And I tell you, it's already there. So go ahead and prepare. And lots of you are helps with these boosters. Okay? Even you did not very good in the exam 1 and the exam 2. Okay, let's talk about these questions. You don't have to do these red ones. These red ones basically will be in the final exam. We're going to mention these later, so you don't have to do. Okay, let's talk about these. A non-protein component of an enzyme that is loosely attached to the protein component is referred as what? Okay, loosely attached and closely attached. What is loosely attached? We call it a coenzyme. Is that right? Closely attached, we call it a prostatic group. If you forget about this question, let's say you forgot about it, and then you go back to look at your slides. That's what these questions for. Is not for you to remember it, is for you to know what's going on there. So if you forget about it, you're going to go back to see the enzyme part, which is right here. Prostatic group firmly attached, coenzyme loosely attached. So this is a slide related to that question. That's what you're going to study. Hollow enzyme is apple enzyme and cofactor combined. Thermodynamic law states physical and chemical processes occur in such a way that randomness disorder increases to a maximum. First of all, there is no third and the fourth law. We only talk about first and second, and it's not the first, and that will be the second, because entropy will be increased. Okay, so that's easy. Question number, answer will be B. Where it is, go back to look at your slides. That would, it's actually not really easy, and the concept is easy, but it's really, explanation is difficult. Look at here, that's the second law. Amount of disorder is increased. Right here, exactly the same words. Can you see it? So then you should click here, and that relates to question number 2. That's what you study. Okay, this question a little bit difficult. Below are the components used by cyanobacillus to make energy from oxygen and iron. And you have cytochrome A, cytochrome C, ferric acids, oxygen, rustocyanin. It's a color pigments. Please put the components in the correct order in the membrane and show electron-proton flow with arrows. Okay, how to do this? You always should remember the higher voltage, which means it's possibly is an electron donor, and the lower voltage is an electron acceptor. So if you don't know how to do this question, you need to go back to C right here. Okay, go ahead to see it. Oxygen 0.82. The electron transport chain is always at the bottom, at the last one. And NAD reduced to NADH is minus 0.32. It's very small. So negative or the smaller the voltage value pretend to be an electron donor, and there will be electron acceptor. So, what's going to be the flow? So flow will be coming from the lowest one, Fe. Okay, I'll just write here. And go to oxygen, okay? Then you just based on the order. The second one is, should be rustocyanine, followed by cytochrome C, then by cytochrome A, and the last one will be oxygen and the water. So this is actually test to you in a, this is a little bit a difficult question because the concept which is put in a story and the cell bacillus we haven't mentioned, but tell you these conditions let you to draw the flow of electron and proton with arrows. Okay? So this question tests you the concept which is related to the slides is right here, which is two-half reaction. So how you apply this knowledge to them. Okay, so that's number 4. Which of the following process generates the greatest amount of energy? Okay, we have fermentation, substrate level phosphorylation, anabolism, glycolysis, and anaerobic respiration. Okay, first of all, fermentation, how many ATP generate? Only 2. Substrate level phosphorylation, that is hard to say. Anabolism, not generate ATP, they use ATP. Glycolysis, how many ATP generate? 2. So what is that? The answer, anaerobic respiration. Okay, if you don't know, where you should find it? And I said this slide is very important. This is right here. Oh, sorry. Not this one. This should be. And I said this here, the slides is important because that tells you all the calculation. Okay, aerobic respiration, 38 ATP. Fermentation is only 2 ATP because it's coupled with glycolysis. Anaerobic respiration, it is glycolysis and TCA cycle. I would say it's a modified glycolysis and TCA cycle. So they will generate around 36 or less. Somehow between 2 and 38, you can say. That's why these choices, the maximum, the greatest amount of the energy generated is anaerobic respiration. Okay, so that will be the answer. That will be the answer. You should cross out anabolism first because it's not generated, it uses ATP. Glycolysis only 2, fermentation only 2, substrate level phosphorylation we don't even talk about here because it's a concept with phosphorylation, it's not really generate energy. Okay, if you want to really say it's only 1 ATP. Question number 6. Sulfonamides or antibiotics that is functioned by. Okay, this is sulfur drug. And we said it is antibiotics, but it is made. So where are they? This one, you need to go back to see the slides, is right here. It is competitive inhibition. Sulfonamide is right here. The structure is very similar to PABA, benzoic acids. So they can do a competitive inhibition of the enzyme activity. So what is the answer here? Inhibit the synthesis of a vitamin by competing with the normal structure of the active site of an enzyme involved in the vitamin synthesis. Okay? This has to be fixed. Okay? So that's question is C. Okay. Question number 7. The transfer of electrons and hydrogen by a dehydrogenase to an organic molecule as the terminal electron acceptor is called what? This is go back. We need to review what we talked about before. If electron donor and electron acceptor have a different scenario, what's going to happen? If electron donor is organic chemical, electron acceptor also organic chemical. This is what we call fermentation. If the electron donor is organic chemical and the electron acceptor is inorganic chemical, This is requires respiration. And based on the inorganic chemical, if it's oxygen, requires aerobic respiration. And if it is not oxygen, or we say other than oxygen, then there will be anaerobic respiration. This is the key. What we at the beginning of the lecture we talked about that, and this is the best definition to explain this concept. But keep in mind, it's very simple. Like this. This is your study guideline, and that is also your study guideline. That's 4 different scenarios. This is also 4 different scenarios. Then what question we go back to see? The transfer of the electron, the hydrogen by hydrogen, the dehydrogenase, to organic molecule as a terminal electron acceptor is called what? Fermentation. Because we already said, regardless of the donor, if you accept it is organic, fermentation. Okay? Which of the following is the best definition of fermentation? Same thing. The oxidation of a carbohydrate with organic molecules serving as a final electron acceptor. That is the best definition for fermentation. The others, it doesn't talk about fermentation. Okay. Which of the following is used as an electron carrier by living organisms? This one, if you forget, go back to see your slides. If you forget about it, look at these slides. What are the electron carriers? Nicotinic adenine dinucleotides, dinucleotides phosphate, and the fulvine adenine dinucleotides. So it's simple. Okay, the last one we didn't mention NADP because using for Calvin cycle, which is biosynthesis. So we didn't mention that too much. Okay, that's question. So this you could click in, relate to the question is number 9 here. Question number 10. When the end product of a pathway inhibits catalysts of the first step of that pathway, this phenomenon is called. Okay, if you forgot about this, go back to read. What's this? End product could be intermediate, or the end products will be go back to impact the intermediate products or even the substrate A. What is this called? Right on the top, feedback inhibition. So that's what this question for. It's feedback inhibition. This phenomenon is called feedback inhibition. Okay. 11, you don't have to worry. Okay, this question is important. This I have to emphasize, because this one, lots of you didn't get it at all. So we need to talk about that. Okay. Pyruvate become lactic acid. And what happened here is NADH reduced, oxidized, become NAD+. And I said this is important because there is a container you could be carrying electron. That's the reason why fermentation only 2 ATPs generate, but the bacteria can still survive. That's the reason. Okay, first of all, what's the name of this reaction? Name this type of biochemical reaction used by the microorganism. This is easy. This question is only for you to not fail. So that's called fermentation. That's easy, the first question. Second, please name the reduced products, oxidized products, reducing agent, and the oxidized agent. Okay, let's do it. Reducing agent, oxidizing agent. Okay, oxidized product and the reducing reduced the products. Okay, reduced the products. Okay, let's do this. What is oxidized product? Lose electron. What is reduced product? Lactic acid. And we call it pyruvate. Reduced to lactic acid. Okay, what is oxidizing agent? Pyruvate. What is reducing agent? ADH. Is that correct? Yeah, there should be all opposite way. Always remember the reduced product when you go here is oxidizing agent. What's the biological function of NAD in this reaction? That's the electron carrier. To recycle. Is that right? Electron carrier have to be recycled. So biological function. That's a very typical question for the fermentation. Name one food product produced by this reaction and name the microorganism related to that. You know that cheese, lactobacillus. And if you don't know, there's a lots of answer could be here. Okay, lots of the answer you can pick from right here. The whole picture you can see it. You can pick anyway you like. Probionic acids for Swiss cheese. Lactobacillus for like cheese, cheddar cheese, soy sauce, Saccharomyces for beer, Clostridium for acetone, acetic acids for E. coli for vinegar. That's all you can pick. Which of the following can be used as an electron acceptor during anaerobic respiration? Okay, now you go back to see the anaerobic respiration. That's how I say it. It's the key. Not oxygen, other than oxygen. So, all of these. Nitrogen, sulfate, carbon dioxide, all of the choices. If you don't know the answer, let's go back to see. The answer is right here. Anaerobic respiration, look at the picture. These are all listed electron acceptor. So this question corresponding to these slides. I didn't randomly pick these questions. It's all reasonable. There are the slides most likely corresponding to that. That's why you can study. Okay, that is the electron acceptor corresponding to that question. So that's all the answers. So that's A, B, C. That's going to be D, all the choices. Which of the following is not true regarding ATP synthesis? Oh, that's a little bit difficult because we didn't talk too much. They require protein motive force to make ATP. Yes, that's correct. They span the inner membrane of mitochondria. Let's see. The proton flow is outward during the ATP synthesis. The subunits of ATP synthesis are undergoing conformation change during ATP production. Which one is not correct? It is C. It is not outward, it's inward because that's called the proton motive force. If you don't know about this, we need to go back to C, chemosmosis. That question is for chemosmosis to test you. There is a couple of the choices of that question. We didn't mention detail, but there is one thing for sure. This is you have to come back. Is that right? When the ATP synthesis, that has to come back. Because that proton motive force is activates the ATP synthesis, generate ATP, and coupled with flagellar rotation. That's the chemosmosis. Okay? So it's outward during ATP synthesis. That is not correct. Because during the ATP synthesis, it is actually, we are talking about, is right, the dashed line on the right side. At the beginning, it's go out, that's the left side. Okay? So that's a question. The hypothesis that proton motive force drives ATP synthesis is called, that's easy, right on the top, chemosmosis. This is what you're going to study. When you study, don't forget the big title on the top. Sometimes that's the answer of the question. And lots of you directly to see, oh, then you're confused. Don't forget. The bold letter at the top is always you need to keep in mind. So sometimes I give you an easy question, then you say think it's difficult. It actually is right there. You can read. That's chemosmosis. So keep in mind, okay? organisms that form lactate, ethanol, and carbon dioxide as end products of glucose fermentation are called, and this is we mentioned in the class, but we didn't emphasize, fermentation there is a two. For example, pyruvate generates lactic acids. What is called? Hormone fermentation. If they generate the other chemicals other than lactic acids, we call it hetero fermentation. Okay? That's like lactate, ethanol, carbon dioxide, we call it hetero fermentation. Okay, we don't need to emphasize about this guy, is that right? Calculate total theoretical ATP, yield aerobic respiration. Look at my video there. Okay, some of you never read it. Then they come back to say, where are they? Let me remind you. This I put on there is for extra points aerobic respiration ATP's calculation. And I said, if you don't need extra points, because it's not a biochemistry class, you need to know where happened is ATP generated. And you only need to know approximately the location. You don't even need to know the chemical, because it's not a biochemistry class. Assuming you're a freshman, you don't know anything about biochemistry. But most of you know, that's why we offer you the extra points. So that is for aerobic respiration ATP calculations right there. Because I know some of you never read it. Start to study maybe tomorrow evening. And then, where is the side chain? Is right here. That is a video, a little bit short, 15 minutes. We talk about the amino acid side chain and also the ATGCU, the structure. So those two videos definitely you should look, okay? Because we gave you the opportunity for extra points. So if this guy, I'm not going to emphasize here. Where are the slides? Slides right here and right here. Now, some of you confusing. Said, how come you have here is 32, you have here is 38? Because there are two rules. Remember, we talked about, this is the new one, which is you using NAD reduced to NADH2 is 2.5 ATPs, and FAD reduced to FADH2 is 1.5 ATPs. So that's why the calculation is 32. Here we use the old rules, which is 3 and 2. I would encourage you to use the old rules because it's easy. But if you already been training with your biochemistry faculty for 32, that's okay. Only one rule, not to combine. Okay? Only one set of the rules. That's why I always keep this one, because from the mass calculation, this is easy to do. Some of the textbook even start to say FAD reduced to FADH2 is only 1 ATP. But that's some noise there. So we just let you have these two. It's the same thing like we talk about 20 amino acids, lots of textbooks start to talk about 21. I mean, just some noise there. So I just don't want to emphasize that too much. If yeast, just quiet a little bit. Okay, if you have a question, just to have your hands on. If yeast metabolized one molecule of glucose to ethyl alcohol in the absence of oxygen, how many net molecules of ATP would be obtained? This is a very good question because you can guess. But the answer is simple. Yeast going through in the absence of oxygen generated alcohol is fermentation. Does not matter how complicated the fermentation, how many ATP generated. It's only 2. So the answer is 2. This is the question a little bit difficult because you need to know the story here is talking about is fermentation, is hetero fermentation, but fermentation, no matter how complicated, is only 2 ATP generated. It is from glycolysis, which is coupled with glycolysis. Okay, so the answer is C, 2 ATPs. Okay, how are aerobic respiration and anaerobic respiration similar? Go back, right here. That's why I said this is a couple of lines. It's important. They are using inorganic molecule as a final electron acceptor. So what's the answer here? Both use inorganic molecule as a terminal electron acceptor. Now you need to be asking me where it is. You need to go back. If you really wanted to see where on the slides, you need to go back. It is actually right here. But it doesn't really recognize by you because it's kind of confusing. There's a figure there, there is a letter there. What is aerobic respiration? Final electron acceptor is oxygen. What is anaerobic respiration? Final electron acceptor is different exogenous acceptor such as, which means other than oxygen. Okay, so that slide, which is actually the same thing what I wrote here. But if you remember here, that's the same thing for that slide. Okay, that's good. Probionic bacteria makes probionic acid, acetic acids, and carbon dioxide in the Swiss cheese. These products resulted from A, B, C, D. Well, this is easy. You already know it's fermentation. If you forget, right there. Is that right? Fermentation. Is it related to that? Carbon dioxide, the probionic acids have Swiss cheese, those holes are carbon dioxide. So that's fermentation. Okay, this is a question we gave the students a long time ago to do. Citric acid cycle. And this is the one, is a very good question. If you don't know chemicals, we wrote for you. Are there oxidative phosphorylation in this cycle? Please label it. Where are they? What means oxidative phosphorylation? Which means they relate to the electron transport chain. So in other words, every time when you see NAD plus reduced to NADH, FAD reduced to FADH2, that is oxidative phosphorylation. So you should label right here, label right here, right here, right here, and right here. Okay, that's oxidative phosphorylation. Is there a substrate level phosphorylation? Please label. Well, right here. GTP, generated from GTP is substrate level phosphorylation. Where is the place the transition step? Is on the top. Pyruvate become acetyl-CoA. That's called the transition step. Define metabolic precursor. Okay, you don't have to know this. We didn't mention this semester. Name one chemical in this cycle related to biosynthesis protein and fatty acids. Alpha ketoglutarate. We didn't mention, but if you learn biochemistry, you know alpha ketoglutarate is a magical stuff. They go everywhere. Okay, that's why citric acid cycle is important, because when you go here, it goes everywhere. Okay, you don't have to worry about that question. In aerobic conditions, it takes sugar to produce the same amount of ATP when compared to the anaerobic conditions. More or less. Of course less. Is that right? Which of the following can be used as an electron acceptor during anaerobic respiration? Same thing. Go back to see the concept. The inorganic chemical other than oxygen could be nitrate, sulfate, and carbon dioxide, all of the choices. List the central metabolic pathway. This is I didn't mention. This should be glycolysis, TCA cycle, and the pentose phosphates. Fermentation is not, but it's okay. We didn't emphasize this. Okay, what is the difference between first and second dynamic law? Oh, it's easy. You just go back. Why it is we based on extra points? Because this needs to write. So we gave you the extra points. First, energy never created, never generated, or only transferred. Second, disorder amount is increased. Okay? Who proposed the chemosmosis theory? Peter Mitchell, a British scientist. Name one final product generated from pentose phosphate pathway. What is that? And I said this one I didn't talk too much. NADPH should be. Right here. Produce NADPH, which is needed for biosynthesis. and it's a precursor material for ribose, and basically it's for DNA and RNA. Okay, that's phosphate, pentose phosphates, and I always say it will be a little bit precise, because otherwise it goes forever, you know. You don't have to do that one, so that's done. Okay, next set. Next set of the question, let's look at the second one. Okay, this is genetics part. Some question easy, some more difficult. First thing is easy. Two strands of a DNA molecule are what? Anti-parallel. They are rounding opposite direction. If you still don't know, let's say the first time, you never take biochemistry class, so we go back, then we're going to have to see the slides, which is slides number 3, I believe, right here. So if you don't know about this one, I will say you may have trouble, but that's okay. You still have time to study anti-parallel, rounding opposite direction. Is that right? Glycosidic bonds, to combine them, phosphodiester bonds connected with them, hydrogen bonds with each other. Okay. A consensus sequence in the untranslated leader sequence of a prokaryote mRNA, which serves as a ribosome bonding site, is called, what is this called? What we need. Is that called SD sequence? Shard-Lugano sequence? Because that has to be recognized by 16 sRNA, then you initiate the transcription. Okay, number 3. Amino-acyl tRNA synthesis categorized covalent addition of, if you don't know about this one, a little difficult. If you don't know about this one, let's find the slides for you. Okay, let's go to see this guy. This is what I draw on the blackboard, but you may forgot about it, that's okay. Amino-acyl, what's that? Go here. What is this? Formyl methionine tRNA. Is that right? Okay. Covalent addition of, yes. That's the first. That's the first one, and you also could be see right here. It's an amino acids. Okay, you can write formyl acyl methionine. You also can write amino acid. That's both are fine, because I know you understand about that. Okay, attach amino acid. Does the answer is it? The generic information in DNA is divided into units, which is called what? What is it called? Gene, yes. So not all the DNA is gene, is that right? Only half genetic information is called gene. That's good. True or false? The basic difference between RNA and the DNA residue in their sugar and pyrimidine bases. RNA has ribose and uracil, but DNA have deoxyribose and thymine. Is that correct or true or not? Yeah, it's true. Which of the following is not a pyrimidine? Which one is not a pyrimidine? Yeah, adenine. Adenine is what? Purine, is that right? Yeah. Which of the following is not a complementary base usually from DNA? A and T, G and C, A and U. Which one? Yeah, A and U, because this is for RNA. That's we already talk about structure, so it's not difficult. So that's RNA, because here asks is DNA, the DNA molecule. Which of the following is true about the structure of DNA? Purine and pyrimidine bases are attached to the 1' carbon of the deoxyribose sugar. Yes, we talk about the one first, purine and pyrimidine. That's why we got a structure there. Purine and pyrimidine bases are joined by phosphodiester bonds. Is that correct? True or not? Yeah. Adjacent bases are stacked on top of each other. One base pair every 0.34 nanometer. I mentioned about that. So, all of the choices, is that right? Okay. Which of the following nitrogenous bases is usually found in RNA, not in DNA? That's easy. Uracil. When DNA molecule is replicated, the daughter molecules contain one strand of parental DNA and one strand of newly synthesized DNA. This is called semi-conservative replication. And we have an experiment to verify that, which is called Matt Medelson and Frank Starr's experiments using N-nitrogen-15. It's an isotope to do it. So one shape part of the DNA molecule where the actual replication process occurred is called the replication fork. So remember, that's called the replication fork. Right here. See? That's what I said. Go ahead and read on the top. Components of a replication fork. Okay, you focus on these small stuff, but look at the title. Components of a replication fork. So always remember that's replication fork because DNA of the bacteria is usually circular. And for the human being and the animals and the yeast-standard modes is linear most of the time. During replication of the two strands of DNA molecule are unwound from one to another, the enzymes is called what? Helicase, is that right? There's no unwoundednesses. Okay. That's a silly name. You don't even have to do it. Nobody is going to generate like that, those type of the name. So you want to pick B. Okay, it's going to be C, helicase. Then if you say you don't know, then we go back to see here. What is the helicase for? Let's find it. Do we have helicase right here? Maybe we don't have it here. DNA B. Okay, yeah, DNA B. DNA B is a helicase. Right here. Okay. The coding sequence in the DNA is normally continuous. That is not interrupted by non-coding sequence. It's a little bit difficult. So we mentioned the coding sequence of DNA and RNA. What's the difference? There is an exon and an intron for prokaryotes. So what is this? What is the difference? Plants, mammals, most insects, they are eukaryotes. They have intron and exon. The intron have to cut it and the exon have to connect it. That is right after transcription, before translation. But bacteria doesn't have that problem, so we call it a prokaryotes. Okay? The other name for bacteria, we call it a prokaryotes, so that's what it tested here. The region at which the DNA, RNA polymerase binding to is promoter. Okay, this is the one we will mention again. The binding to is called a promoter. This is the important slide you should know. The promoter, a very conservative sequence, is combined with the RNA polymerase. That is what a promoter looks like. And I know it is a little bit different from what you learn in your biochemistry class, because in your biochemistry class you talk about is eukaryotes. This is for bacteria. Okay? So a little bit difference. The unexpressed region of a split genes are called intron, is that right? Which is called spliceosome to cut it. So intron. Pyramidal box of E. coli. What is a pyramidal box? Is centered approximately 10 base pair upstream of the starting site of transcription. If you look at here, that's called a pyramidal box. It's -10 sequence. Why we call it -10? Because that is approximately 10 base pair upstream. So here you see the left side is upstream, this side is downstream. Okay, that's why we use minus to do it. The genetic code is said to be because more than one codon will specify a particular amino acid. This is called degenerate. Okay, that's a redundancy actually. It's called degenerate. Which of the following is not a termination code? What are the termination codes? UAA, UAG, and UGA. Of course, AUG is not because it's replaced by methionine. That's always the first one. And in eukaryotes, it's methionine. In prokaryotes, bacteria, it's formyl methiano. You can say formyl methionine. Okay. Molecules delivery amino acids to ribosome during translation. Which one? Transfer RNA. Okay? That's carry on amino acids. It has an anti-codon. If you don't know, then we can go back to read. Right here. Transfer RNA. The slides there. Glutamic acids, tyrosine, serine, alanine. See, that's anti-codon region. In ribosomes can attach to the mRNA and begin translation even though the transcription has not been complicated. Which one? Prokaryotes, is that right? Because eukaryotes from the protozoan, some plant cell, eukaryotes, it's the same thing. Translation has to be done, finish the transcription, and also some modification. But prokaryotes you don't have to do. You can do it continuously. The reaction sequentially links adjacent amino acids to growing polypeptide. That's called a transpeptidation. Is that right? Transpeptidation rely on 23 sRNA in the 50 S subunits. Okay. What are the biological function of DNA polymerase 1 and polymerase 3? Of course, number one, polymerization. That's simple. 5' to 3' polymerization. What's the function of DNA polymerase 1? Cut Okazaki segments. Then what the other function of the DNA polymerase 3? Proofreading, remember? Fidelity issue, how we solve it? The number one step is DNA polymerase 3. So, here we want to emphasize it. This is something key. People doesn't really pay much attention.